Hi everyone, Assalamualaikum. In this video, I would like to share the simplest yet very important topic in biology, cell structure and its function. My main focus in this video is each component inside the cell and its specific function. Feel free referring to any textbook or any reference book for further understanding, so that at the end of this video, you should be able to state the main function of each component inside an animal and a plant cell. Ready? Let's go. Have you all ever wondered how your cells work? How a microscopic structure, a fundamental building block of all living organisms, plays an important role in making sure the survival of a living organism? The answer is they rely on each other, working together to meet the needs of the cell. Inside the tiny structure, there are a lot of components, also known as organelles, playing different roles, working together, making sure that the cell is in the best condition to stay functional. Basically, in this video, I will compare a cell to a school, an example that is closer to you so that you can understand the function of each component clearly. A typical school should contain the school compound, the classroom, the corridor, the canteen, the teachers, and of course the students. Teacher receives informations and instructions from the headmasters, getting all the knowledge and input from the books or other materials, walk into the classroom to teach and make sure the students excel in their own way. While others in school play different roles to make sure that the school can function efficiently, just like a cell. Different parts in the cells play different roles so that the cell can function efficiently. Picture this in your brain, a cell and a school. Imagine yourself as a student coming into the school. You will first pass through the gates, walk along the corridors, into the classroom. The teacher came, lesson started, you went for your recess, for refreshments, sit for exams, getting good results, and eventually graduates from the school. Or imagine yourself as a substance trying to diffuse into the cell. You will first pass through the plasma membrane, floating in the cytoplasm, waiting for instruction from the nucleus, carry out tasks, getting worn out, digested, and eventually excreted out from the cell. Did you get the image? So, we should start with entering the school or a cell. You will first reach the main gate of the school. There will be park guard on duty, and the gate act as a border that separates the school compound with neighborhood environment. And the park guard will watch over every movement in and out of the school. Just like the plasma membrane of a cell, it controls the movement of substance in and out of the cell. And at the same time, it acts as a barrier that separates the internal environment of the cell with the external environment. So, plasma membrane is like the gate of the school. It controls the movement in and out, and at the same time act as the barrier that separate the internal environment with the external environment. As you enter the school, the first thing that you will step on is the school compound, where all the busy teachers, students, the classroom, the buildings, the canteens, and other facilities are located. This is where all the magic happens, which is like the cytoplasm of a cell where all the cell components are located. And cytoplasm also serves as a site for all biochemical reactions. Cytoplasm is like the school compound, the place where everything happens. All the teachers and other departments in the schools are coordinated and receives instruction from the headmaster. He or she will give specific instructions to the teachers or departments as how to operate and what results to achieve. Likewise, in a cell, all the activities are controlled by the nucleus. The nucleus contains DNA, the specific instructions needed by the cells to synthesize the protein. You will learn protein synthesis in the next chapter. Nucleus is like the headmaster, where they control everything. The instructions and orders from the headmaster are received by the teachers or workers to produce desired results. This is like the ribosome in a cell that is responsible to synthesis protein needed by the body or the cell. The ribosomes may present on the rough endoplasmic reticulum or they can exist freely in the cytoplasm. Exactly like teachers. They may be in the classroom teaching, walking around the school compound, or maybe like the clerks who are loyal by their desk doing paperwork. In a way, ribosomes are like teachers and workers. They are responsible in producing results or desired products. With the informations or instructions from the headmaster, combined with the knowledge from the books or other materials, the teacher walks through the corridor into the classroom. And the students may also use the corridor to walk around the school compound. In a cell, instead of having a corridor as a transport medium, a cell consists of endoplasmic reticulum. Endoplasmic reticulum act as a transport system that transport protein synthesized by the ribosomes to the Golgi apparatus. Endoplasmic reticulum is like the school corridor, where it helps the teachers move from one class into another class, or the students from one location to another location. Or it can be like a travel letter in the airport where it transports you from one point to another point. Upon arriving at the classroom, the informations or input or knowledge that the teacher has are taught to the students. The teaching may involve simplifying notes, doing presentations, doing group works, or even extra exercises as to enhance students' understanding, allowing them to get good results during examinations and eventually graduates from the school. The same thing also happened at the Golgi apparatus. Proteins and carbohydrates from the transport vesicle are processed, modified, packed, and transported internally or externally of the cell. These transport vesicles originated from the endoplasmic reticulum, buds off, and carry protein and carbohydrate to the Golgi apparatus. Golgi apparatus is like the classroom, the place where the modifications and the packaging are done before the product is transferred out. All the functions in the school are made possible because the canteen constantly provide the food for everyone in the school so that everyone have enough energy to work and function at maximum level. Inside a cell, mitochondrion act as a site that generates energy in the form of ATP molecule. These ATP molecules are used by the cells to carry out their functions. Mitochondrion is like the canteen. It provides energy so that all the process can occur at maximum level. Lastly, another important figure in the school that making sure everything is maintained in a comfortable environment are janitors who clean up after the school. In a cell, this role is done by the lysosome. Lysosome hydrolyze complex organic molecule, break down cellular waste or burn out organelles. In a way, lysosomes are like janitors. They keep the environmental comfortable so that all the process can occur efficiently. As an addition, the plant cells contain extra components compared to the animal cells. For example, chloroplast. Chloroplast functions in trapping sunlight and convert it into chemical energy during photosynthesis, just like solar panels that are installed on the roof of some schools or even houses. These solar panels absorb sunlight and convert it into electrical energy to power up the building. In a way, chloroplast is like the solar panel. It traps sunlight and converts it into another form of energy. And the plant cells also have a large central vacuole that stores water, mineral salts, and at the same time maintain the turgidity of the plant cells. In a school, there is a water tank that stores water for the usage of the whole school community. Vacuole is like the water tank, it stores water. So, that is everything that you should know, understand, and remember about a cell. Plasma membrane is similar to the gate. Cytoplasm is similar to the school compound. Nucleus is similar to the headmaster. Ribosomes similar to the teachers. Mitochondrion similar to the canteen. Endoplasmic reticulum is similar to the corridors. Golgi apparatus is similar to the classroom. Lysosomes is like the janitors. Chloroplast can be compared to the solar panel, and large central vacuole in a plant cell is similar to the water tank. Next time, when you are having difficulties remembering the function of each cell components, try to relate it to your school, or you can remember my explanation. Hopefully, it will be helpful. As to test your understanding about this topic, help yourself by giving a click on the link in the description below. Answer a few simple questions related to this topic. So, tips of the day. When you are stuck at certain biology concepts, try relate it to your daily life experience. Make an analogy. Trust me, it will help you understand the concept better. But remember to check the analogy that you make with your teacher to avoid any misconceptions. That's all for today. Remember to stay safe, maintain physical distancing. Together, we can win against COVID-19. Thank you for watching my video. Have fun learning biology.
Hi, good day everyone. My name is Cikgu Amalini. I am a biology teacher at SMK Taman Bukit Maluri. Today, I will explain about the intracellular and extracellular functions of the enzymes and the form reaction of the extracellular enzymes. The prior knowledge that you should have now is the components in the cells. So in the cells, the components are nucleus, cytoplasm, ribosome, rough endoplasmic reticulum, smooth endoplasmic reticulum, cell wall, the plasma membrane, mitochondria, and also we have the lysosome. But which one of these components are involved in the production of the enzymes? Stay tuned until the end of this video. An enzyme is a biological catalyst that speeds up the rate of reactions. Almost all the enzyme is made up of protein and is produced inside the cells. There are two types of enzymes, which are intracellular and extracellular enzymes. Intracellular enzymes is the enzymes that is synthesized inside the cells for its own use. For example, enzyme hexokinase is the enzyme that is produced inside the cell and is used during cellular respirations. By the other hand, extracellular enzymes is the enzymes that is produced by the cell and will be secreted for the use of the other cells. For example, trypsin is produced by the pancreatic cell and will be secreted to duodenum for the breakdown of the polypeptide. Next, I will explain about the production of extracellular enzymes. In the previous video with Cikgu Amalina, you've learned about cellular components and their functions. Which of the components are involved in the production of extracellular enzymes? There are ribosomes, rough endoplasmic reticulum, transport vesicles, Golgi apparatus, secretory vesicles, and also plasma membranes. But how are these components involved in the production of extracellular? So ribosome is the first components that involved in the production of extracellular enzymes. Ribosome is the site of the protein synthesis. Secondly, protein that is synthesized by the ribosome will enter the lumen of the rough endoplasmic reticulum. Rough endoplasmic reticulum is function as a transportation of the proteins. So through the rough endoplasmic reticulum, Protein is transported until it reaches the end of the rough endoplasmic reticulums. Once it reaches the end, the membrane of the rough endoplasmic reticulum will burst off and will form a transport vesicles. Later, the transport vesicle that contain proteins will move toward the Golgi apparatus and then fuse with it. You know that the Golgi apparatus is functioned to process, modify, pack, and transport proteins. So here, the Golgi apparatus will modify the proteins into the specific enzymes. These enzymes will be secreted in the secretory vesicles. They are formed from the tip of the Golgi apparatus. Secretory vesicle will move toward the plasma membrane. After it reaches the plasma membrane, the secretory vesicle will fuse with the plasma membrane to secrete this enzyme outside of the cells. Now, this enzyme is already outside of the cells. So that is how the extracellular enzymes is produced. Next, I will explain about the general characteristic of the enzymes. Enzymes are biological catalysts that speed up biochemical reactions. Enzymes act rapidly, and it is needed in small quantities and are reusable. The structure of the enzymes remain unchanged after the end of the reactions. The actions of the enzymes are specific. It only acts with the substrate that can fit in with their active sites. Most of the reactions of the enzymes are reversible. Enzymes activity can be slowed down or stopped by the inhibitors, and also the enzymes can be speed up by the cofactor. Okay everyone, that's all from me, but you have to stay tuned with the video with the next subtopic with the next teacher, that is Cikgu Syaira, that will explain about the factors that affect the functions of the enzymes. Alright, so have fun learning biology. Bye bye.
Hi everyone, I'm Shaira. So next lesson we're going to learn about the mechanism of enzyme action, the lock and key hypothesis. So most enzymes are complex protein that has an 3D structure. This 3D structure has an active site that complement into a specific substrate. The binding of molecule substrate on specific enzyme is like lock and key hypothesis, because the enzyme is represented by a lock and the substrate is represented by a key. Most reactions in the cell require activation energy. So what is the activation energy? The activation energy is the energy needed to break the bond in the substrate molecule before reaction can occur. So enzyme function is lowering the activation energy. By doing so, the rate of biochemical reaction inside the cell is accelerated. Okay, in this diagram shows that the differences between reaction without the enzyme and the reaction with the presence of enzyme. As you can see here, the activation of energy with the presence of enzyme is lower than compared to the activation energy without the presence of enzyme. There are many factors that affect the mechanism of enzyme. The chemical bond in enzyme can be transformed as a result of chemical changes or physical condition. So in this chapter, you're going to learn 4 main factors that affect the mechanism of enzyme. The first factor is temperature. In this figure, it shows the effect of temperature on the rate of biochemical reaction that controlled by enzyme. As you can see, at low temperature, the rate of reaction catalyzed by enzyme is low. When the temperature rises, the kinetic energy of the substrate molecule and enzyme also increases. This increases the frequency of effective collision between the substrate molecule and enzyme molecule. The rate of reaction between enzyme and substrate molecule increase with every rise in temperature of 10 Celsius. The rate of reaction controlled by the enzyme will double until it reaches the optimal temperature. As you can see, number 2 is represented by the optimal temperature. At this optimal temperature, the enzyme reaction is at its maximum. The optimal temperature for enzyme reaction by human body is around 37 degrees Celsius. After reaching the optimal temperature, any further increase in temperature will reduce the enzyme activity until it stops at 60 degrees Celsius. Why? Because at this temperature, the enzyme becomes denatured as the chemical bond in the enzyme molecule break at extreme temperature. The enzymes are unable to retain the 3D form. The active site of enzyme change, so the substrate does not complement the active site of enzyme anymore. As you can see here, this is the molecule of a normal enzyme and is the molecule of denatured enzyme. The 3D structure is changed. Factors number 2 is pH. This is the graph diagram showing the effect of pH on the enzyme activities of pepsin, salivary amylase and trypsin. The enzyme activity is influenced by the pH of the surrounding solution. Generally, all enzymes react most effectively at their optimal pH. Most enzymes are most active in the range of between pH 6 and 8. For example, the salivary amylase works at pH 6.8 and 7. However, there are some exceptions. For example, we have pepsin enzyme in our stomach that act the optimal pH range between 1.5 and 2.5. The trypsin enzyme in our duodenum, on the other hand, only works well in alkaline medium at a pH around 8.5. It is because the change in pH value change the charge of hydrogen ion of the active site of enzyme and the substrate surface. As a result, the enzyme-substrate complex cannot be formed. When the pH of the environment returns to the optimal level, the charge of the active site will be restored. The enzyme will return to function as a normal. The extreme exchange in the pH value will break the structural chemistry bond and change the active site of enzyme. Okay, next, let's move on into substrate concentration. As you can see in the figure, this is the effect of substrate concentration on the rate of enzyme reaction. If the concentration of enzyme is fixed while the concentration of substrate is increased, the reaction is controlled by enzyme also will increase, leading to an increase in the product created. When substrate concentration increases, the opportunity for an effective collision between the substrate and enzyme molecule also increase. The reaction rate continues to increase until the maximum level. The rate of reaction in the maximum level is constant. At this maximum level, the concentration of enzyme becomes a limiting factor. The reaction rate can only increase when the concentration in enzyme increases. After reaching the maximum level, all the active site of enzyme are saturated with the substrate and are involved in the catalytic reaction. And lastly is the enzyme concentration. As you can see in the graph or figure, they have the effect of enzyme concentration on the enzyme reaction rate. When the concentration of enzyme increase, the rate of enzyme reaction will increase because of the presence of more active site. They are ready for catalytic action. Next, if the concentration of enzyme in one reaction is double, the amount of substrate converted to product per unit also double with the condition that there are excess supply of substrate. At this rate, the concentration of the substrate becomes the limiting factor. The rate of reaction can only be increased by adding more substrate. So, there is 4 factors that affect the mechanism of enzyme: temperature, pH, substrate concentration, and enzyme concentration. To check your understanding, please click on the link in the description below and answer a few questions related to the topic that we discussed earlier. Okay, thanks for watching. Bye-bye.